All right, so I'm happy to introduce our second contributed talk of this session by Khang from the University of Oslo.
Thank you very much for inviting me here today. I'm really excited to share our current research at the Computational and System Immunology Lab at the University of Oslo. So first, I would like to start with a question that we want to ask. So as you all know, when we do AIRRC6, we can examine the BCR repertoire. And when we want to examine the serum antibodies, we can do antibody mass spec to know the sequence of those antibodies. But what we don't know here is how much is the overlap between these two sections of the antibody repertoire. And previous research have not found correlation between the abundance of BCRs and serum antibodies. And therefore, the link between genomic and proteomic antibody diversity is still unclear. That is why we are doing our research here, that the main aim of our project here is to integrate genomic and proteomic antibody data together. So we do this by first, from a human volunteer, we collect blood sample and we isolate and sequence B cells in bulk and in single cell sequencing. We have the full VDJ sequence, the BCR sequences, and we use those to build a custom reference database. So why do we do this? Because from the same blood sample, we will isolate serum antibodies, digest them with enzymes, and cut them into little pieces so that the mass spectrometry can sequence the antibody sequences of these antibodies. So because they are in short peptides, we need to map them back to their original reference in the BCRs. So our main work here is to integrate all these processes together. But one thing here is that because antibody mass spec is still quite underexplored, so first we want to know how it works. So what is the lowest concentration that we can detect an antibody and how reproducible it is, and other factors related to technical questions about the performance of mass spec. So we are also doing a benchmarking work on this project. So first, with the data from the integrated analysis, so we isolated serum antibodies from all the isotypes from the serum, and we have all of the mass spec peptides. As you can see on the first figure, we have more than 1,000 different mass spec peptides, but when we map them to the reference, we only have a small fraction of them, so 22, that can map to a CDRH sequence of an antibody. Why we do this? Because this is the part that we're interested in, because it can identify where this peptide comes from. So from these 22 antibodies, when regarding for the method that from the references from, we can see that most of them map to a bulk-seq reference, which is unsurprising because it has a much higher throughput. But some of them map to the single cell reference. And this is interesting because with the single cell reference, with a peptide from mass spec, we can potentially link to both chains of an antibody from a peptide. And one other thing is that when we digest the antibodies, we use multiple enzyme treatments. So it will cut at different places. We do this because we want to maximize the coverage of the antibody peptides. And we can see in the figure that all these 4 treatments, they contributed quite equally to the detection of CDRH peptides. And one other thing is that even though a peptide can map to a CDRH, it doesn't mean that it maps exclusively to one CDRH. And we can see that only 14 out of 22 can map to a unique CDRH. The rest is mapped to a more like common sequences of the CDRH. So those are not quite so useful to identify a clonotype. So regarding that, now we go to the BCR references that we built earlier, and we check the mass spec peptide with that. So I have an example here. First, with the green side, you can see that this peptide is quite long and it overlaps quite nicely with the CDRH region. So from here, we can check with the reference for the BCR reference. And from there, we can have the original VDJ sequence from this mass spec peptide. But on the right, this peptide is quite short, and unfortunately, it mapped with a region with multiple different CDRHs. So in this case, we don't quite sure where these peptides come from. So this illustrates the fact that there are still a lot of improvement to be made in both improving the sequencing depth, so we have the most wide coverage reference as possible, and also improvement in the performance of antibody mass spectrometry. So in order to address that second part, the mass spec part, we are also doing a benchmarking experiment where we have 6 monoclonal antibodies, which we already know the sequence prior, and we put them in multiple different combinations with high concentration and low concentration of antibodies, and with or without the presence of background antibodies to create noise. And we also try different enzyme digestion strategies so that we have a wider coverage. And finally, we have 4 biological replicates to test the reproducibility of this method. So first thing is a hunch was correct because biological replicates are quite necessary to increase the detection coverage. What I mean by that is, as you can see in this Venn diagram, in the center, you can see regarding to the CDR3 related peptide, most of the peptide, they are quite reproducible from one run to the next. But on the outskirts, you can see that there are cases where there are peptides that map to only one of the 4 replicates. So we can see that every time we run the mass spec with the same samples, we detect something new. So it means that replicates are quite necessary for a more comprehensive look. So another thing regarding the limit of detection, so far, results have shown that we can reliably identify a CDR3 peptide at an input of 100 nanograms, that is with the presence of serum IgG, and 10 nanograms without the presence of background antibodies. And one question that arises from this is that, can the mass spec signal be a good indicator of the antibody concentration? So we mapped the antibody peptides that on the y-axis is the mass spec signal, and on the x-axis is the ratio, the concentration ratios between the monoclonal antibodies. And if it's going as expected, then it should follow a diagonal line. So we can see that for some of them, they follow quite closely up to a ratio around 1 to 100. But if higher than that, then it kind of drops off. And we can also see that it works better for some antibodies, but not others, and for some enzymes combination, but not others. So we can see that this process is quite dependent on the specific interaction between the antibody sequence and the digestion enzyme. Also, in summary, for the first part in the integrated pipeline, we now know that detecting and recovering antibodies full VGA sequence using mass spec is possible. However, there are a lot of challenges that remain and a lot of improvements to be made here. And secondly, with regard to the performance of mass spec, it is highly dependent on both internal factors such as the antibody sequence and external factors like the choice of the enzyme we use, the presence of background noise in the sample, and the performance and sensitivity of the mass spec system. So for that, to sum up, recently we have published a review that outlined the promises and challenges in applying mass spectrometry to antibody repertoire research. And in the future, we would like to expand this by focusing this method on specific B-cell subsets and couple them with mass spec data to answer more biological questions. And we also want to compare the performance of mass spec using reconstructing the peptide sequence with the reference and without the reference. And we want to relate the result of our benchmarking experiment to the biological distribution of antibodies. And finally, we want to test this integrated data analysis workflow with data sets that have the sequence from bulk seq, single cell BCR6, and preferably with mass spec data as well. So if any of you have any kind of data is resembles this, we are really happy to collaborate with you on integrating this workflow. So finally, thank you very much for listening. I would like to thank the project team at the University of Oslo and the University Hospital. and everyone else in the CSI lab. Thank you very much for listening. My poster is also here, so if you want to know more about this, please come by and have a chat. Thank you.
Maybe I'll just start off with a question, which is in terms of your, actually, in terms of the bulk sequencing that you did you do any testing? Do you know if you've saturated the bulk sequencing, or how deep did you actually go, and how deep do you think you would have to go in order to saturate, to map everything?
Well, we haven't done testing to see like how saturated it was, but I think that because of the limits of like currently we only have like using the MiSeq because we want to have the full length sequence. So we think we can go deeper in that regards, because I think that the low number of mass spec peptides we have is probably one part due to like we don't have enough depth in the BCR sequencing. So basically our reference is not quite big enough yet, but we would like to make it bigger and more expansive.
I have two questions. So the first one is, have you looked at plasmablasts or the secreting cells rearrangement and compare this? Because maybe what you're looking by mass spec is diluted in these massive repertoires that you can get from the whole B cell population. So did you look at more a subset of cells?
Yes, so that is exactly what we want to do next. So for that, we want to focus on the B cell sequence of the plasmablast and the secreted antibodies, because that would be a more narrow search, and I think it would reveal a lot more overlap than just unsorted B cells. Yes, it's definitely what we are thinking of.
And the second question is the limitation also on the mass spec side, would it be also associated with the modification, acetylation, or other protein modification of the antibodies? So depending on the antibodies, you have some patterns. Did you check that in your monoclonal one?
Yes, so I think it would be a really interesting thing to look into. So far, our exploration is quite in its early phases, but in the later phases, we would really like to look into the modification and how it affects the detection of the mass spec peptides. Yes.
Sorry, just a sort of off the wall thing, but I mean, as the only fragments that are useful are the CDR3 fragments. Most of the stuff that you're throwing down the spectrometer is not the CDR3 fragments. Is there any way you could separate them? Can you think of any way that you could just put the CDR3 segments in?
So far, I don't think of any way to do that like specifically for CDR3 yet. But one of the common things that people do when they do mass spec with antibodies is they cleave off the constant region and they focus only on the variable region. We have also tried that, but one thing we found is that during the process of cleaving off that, we lose some of the diversity already. So we don't want to bias our samples too much. Yes.
Thank you.